Hello, my name is Ariel Rokem. I'm at the University of Washington, and I'm one of the core developers of DIPY. And in this video, I'll tell you a little bit about diffusion tensor imaging in DIPY and more generally about diffusion tensor imaging and modeling of diffusion data. So I'll start by defining a few things, by defining what we talk about when we talk about models of diffusion. I'll make a distinction between two different kinds of models, phenomenological models and mechanistic models. And then I'll focus in on the diffusion tensor model, which is a classical model of diffusion. I'll start by explaining this model in 1D, but we'll quickly go to an explanation of this model in 3D, where we model the diffusion in multiple different directions. Next, I'll talk a little bit about the derived quantities from the DTI model and their interpretation. I'll go on to a hands-on example showing you how to use DIPY to fit the diffusion tensor imaging model. And then finally, I'll talk about a few of the advantages and disadvantages of DTI, some of the pitfalls in interpreting the results of DTI. So first of all, just in general, what are models for? Models are used in science as tools for inference. We use them to summarize the data, endow the data with specific meaning and interpretation. They allow us to predict other data when we use machine learning or statistical learning techniques by fitting the model to some data and then predicting some held out data or some other data. And then finally, models allow us to explain our results or formulate a theory about our data. And I'm pointing here also to a great paper by Novikov et al. From 2018 called simply On Modeling, which explains a lot more about these ideas, specifically in diffusion MRI. So as I said, there are two different classes of models that you can think about, just in general. Phenomenological models are, as quoted here from the Stanford Encyclopedia of Philosophy, models that only represent observable properties of their targets and refrain from postulating any hidden mechanisms. These are models that represent the data. Mechanistic models, in contrast, do postulate hidden mechanisms. For example, they might postulate something about the biophysical properties of the tissue or the microstructure within the voxel. They might postulate something about the orientations of nerve fiber populations within a voxel, and so forth. Each class of these models has its own advantages and disadvantages. In general, phenomenological models can be quite ambiguous because they don't tell us much about what the hidden mechanisms are. So different hidden mechanisms can cause changes in the model that are identical or indistinguishable. On the other hand, mechanistic models require making some assumptions about what mechanism causes what change in the signal. So those are the kinds of things that you need to balance when you think about selecting a model for modeling your data. The DTI model is based on this experiment, the pulse gradient spin echo experiment. In this experiment, spin echo experiment is conducted. Here's the pulses for the RF pulses for the spin echo experiment, and then set of pulsed gradients or a pair of pulsed gradients are applied to the sample, and these gradients have a particular direction, they have particular duration and amplitude, and all these parameters that parameterize this are important when we subsequently want to model the signal that arises from the voxel in response to this MRI sequence. So in 1965, Descoteaux and Tanner defined what I'll call the diffusion tensor model, but in one dimension. This is the Descoteaux-Tanner equation. This Descoteaux-Tanner equation tells us how the signal changes as a function of these various parameters of the spin echo, pulse gradient spin echo experiment. And it turns out that it goes as e to the minus some combination of these parameters times d, which is coefficient of diffusivity. And then subsequently people, with Bihanan specifically, said, well, let's summarize all of these things in one number and call that the B value. And that's the B value that we measure in the diffusion MRI sequences that we use nowadays. So this is the model in one dimension. But in fact, we want to model in diffusion MRI differences between situations where diffusion is the same in all directions, what I'm calling here isotropic diffusion here, little water molecules bouncing around maybe in some kind of cavity that is isotropic in its directionality, versus what we call anisotropic diffusion, which is what we have inside of white matter. So inside white matter, the water is restricted to diffuse more readily along the length of these nerve fibers than across the boundaries. We get anisotropic diffusion. Diffusion is more along the length of the axon then across the boundaries of the axon. Now, diffusion MRI data is structured in the following way. We measure multiple times with different B values and different B vectors. And that's what we enter into our modeling process, is the series of data that we have in every voxel. And this is what it looks like in practice. Here, what I've done is I've taken a series of images from a diffusion MRI experiment in 3 orientations, the sagittal, axial, and coronal slices. And what you see is I'm rotating around the gradients, and you can see that the color changes in every voxel. And those differences, changes in the color, is what we want to model in our diffusion modeling. What diffusion tensor imaging does is that it tries to take the measurements in all those different directions and model them as a 3x3 matrix or a rank 2 tensor, where each one of the elements within this rank 2 tensor models the variance or covariance of diffusion in some set of axes. So, for example, this will be the variance of diffusion along the x-axis, the variance of diffusion along the y-axis, and the variance on z-axis. And then there are 3 covariance elements. You'll notice that because of the way that diffusion MRI is measured, the lower triangle of this and the upper triangle of this are actually identical. That's because the covariance xz is the same as the covariance zx. And so our model provides us with this kind of smooth representation of the original data. You can see this is a pretty good model of the original data. So here it is now written out in math. The diffusion tensor that I just showed you is this Q matrix up here. You'll see that this is a form of this Descoteaux equation, but now so we had before e to the minus b d. Now d is a projection of this 3x3 matrix or rank 2 tensor onto a b vector. In this case, the b vector is theta. So now the measurement is dependent on a direction theta and not only on the diffusivity. And so this is the projection of the diffusivity of the tensor onto the vector theta. Here it is rewritten by moving the B0 measurement, the non-diffusion weighted measurement down here. So we get a ratio, and this is the full equation. Now we can take log of both sides of this, and then we get a log of the signal as dependent on the direction divided by the non-diffusion weighted signal is now this linear model of with B and this element is a single number because it's a projection. And so because we've done this trick of dividing by 0 and then taking a log transformation, this can be fit as a optionally weighted linear model, but as a linear model. So that's a really clever trick here, and is due to Peter Basser and his colleagues in their seminal 1994 paper that introduced the method. Now, given this rank 2 matrix, this 3x3 matrix, a rank 2 tensor, a 3x3 matrix, you can do an eigen decomposition of this matrix and get 3 eigenvectors and 3 eigenvalues that correspond to these eigenvectors. And we can take those quantities and use them in inference. For example, we can take the eigenvalues and we can average them, and that we call that the mean diffusivity. We can also compute a weighted variance of the eigenvalues, and that gives us a number between 0 and 1 that tells us how different diffusion is in different directions. We call that the fractional anisotropy. So there's a map of the mean diffusivity based on DTI and a single slice, horizontal slice to the brain. And this is what fractional anisotropy looks like for the same slice. And you can see that there's a bit of a complementary picture here. The mean diffusivity is very low within the white matter and gray matter of the brain, where diffusion is relatively restricted. And it's relatively high inside of the ventricles, where diffusion is, the water can diffuse more readily in multiple directions. However, within the ventricles, fractional anisotropy is rather low, and that's because diffusion is the same in all directions. In contrast, in these white matter bundles, for example, the corpus callosum or large white matter pathways, through the white matter, the fractional anisotropy is high because their diffusion is relatively restricted to some directions relative to the other directions. Finally, we can take the first eigenvector, and that's the principal diffusion direction. So that tells us here encoded with red as the right-left direction, green is anterior-posterior, and blue as superior-inferior. And in every voxel here, the principal diffusion direction vector RGB is then weighted by the fractional anisotropy. So we get more saturation in these parts that have high fractional anisotropy. And we can see, for example, within the corpus callosum, most of the diffusion is in the right-left direction. In contrast, in some of these other pathways, for example, here, there's a big bundle, the corticospinal tract, that goes inferior, superior, and that's why we get this more bluish color. In other parts, we may get more anterior, posterior, so more green colors. Okay, so that's just an introduction to DTI in general. Here's how you run DTI using the command line interface for DIPY. The first thing that you would do is you would fetch some data. In this case, we're fetching an example data set, the Stanford Hardy data set, and we're setting the outdir to be the current working directory where we're working currently. In order to fit DTI, we require that a mask be present. So we are going to use the median Otsu. This is a brain mask or brain extraction algorithm. We run that on the data that has just been downloaded. We give it the volume index 0, and we also say where we would like to put the brain mask, and we put that inside of the directory that was created in the first fetching operation. Then finally, we fit the model. The model takes as input the Hardy data itself. This is the diffusion weighted data, then a set of B values and a set of B vectors that were provided. And then finally, it requires this brain mask that was just generated in the line before. And then we tell it where to put the outputs. And the outputs of this operation are going to be maps of the eigenvalues, the eigenvectors, and then also of a few of these statistics like FA and MD that get saved as separate images. And you can run this on a single file as we ran here, or you can run it on multiple files by using the Unix wildcards here in this call. Okay, so that's how you run DTI and DIPY. A few of the pitfalls of DTI that we should talk about now, because you'll hear more about those in other talks in this workshop, is first the fact that it does not well represent crossing fibers. For example, in the case in which there are two fiber populations within one voxel, the DTI model will represent an average direction. The principal diffusion direction of that voxel will not be the principal diffusion direction of one of the fiber populations or the other. It will be something that is in between those, the average of those. And that can be a problem for tractography. The second problem is that the interpretation of these results, the FA or the MD maps, is quite ambiguous. And for example, FA can change because of many factors. Myelination or demyelination is one of those factors, but the presence of fanning or crossing fibers within the voxel can be another reason that FA can go down, for example. Another effect that might affect FA is the partial volume with other tissue compartments. For example, if there's partial voluming of corticospinal fluid or of gray matter, the FA will go down without any change in the white matter tissue itself. So my advice for you is that you should use DTI, definitely a very useful model and one that well represents the signal, but you should avoid making strong mechanistic claims based on this kind of phenomenological model. For example, you should avoid saying that because two groups have a difference in FA, that difference must be because of differences in myelination or demyelination or things like white matter integrity, because they could be due to other factors as well that affect the signal. Okay, finally, to close this video, I'd just like to thank you all for listening, thank my sources of funding, and with that I close.